Hello, marvelous people. I recently made this YouTube channel to get some ideas in science that interest me. And the way I have it constructed is at first I created 5 videos on topics that I was interested in, but after that, whatever videos got the most views, I'll combine them into making another video. For instance, this video was made because I had a lot of views on my quantum computing video and also a video I made on quantum biology. So this one is kind of comparing biology to a computer and combining both ideas. And that's how I'll continue to make all of my videos so that it's an evolution of ideas that's my interest and also my audience's interest. So biology can be compared to a computer in many ways. As you can see on my left here, you can see the DNA genetic code is kind of like a software code that goes with each chemical that makes up the DNA molecule. A lot of people know of this software code. However, I don't think a lot of people know about many other software codes. For instance, the the morphogenetic codes for structure or morphology, or the bioelectric codes, or epigenetic codes. Those are all codes that overlay on top of the genetic code DNA that everyone's familiar with. But however, I think consciousness and living things pull, and evolution itself pulls from all of these codes to render life and everything that we experience with that. So I think it's actually better if we just compare it in general to a computer code. So to move on, as I said, the DNA is made up of chemicals just like proteins and RNA. So you can think of that as just the hardware, but then the chemical signaling or the signal transduction and the bioelectricity and also the changing in structure that's called morphology. All of those mechanisms, you can actually think of that as the software which makes up all the different codes that are incorporated in the whole biological system to give the biological system behavior, or if it was a computer, applications, and then a user experience or an output if it was a computer. So my favorite way to think about this is to think about it in terms of intrinsically disordered proteins, which make up biomolecular condensates. And you can compare them to Julia sets, which Julia sets are used in computing to make things like graphics. And they're just an iterative function that continually makes a fractal. So fractals can be used to image graphics really well, like waterfalls and stuff, because it looks like nature very well. So this, among a lot of other things that I've talked about on previous videos, is why I think quantum gravity inherently has fractal structure. And that's seen in loop quantum gravity, and in string theory, you have dodecahedron fractal models used for both that can be used to approximate the energy landscapes. So fractals are good to model things in nature, and they're also good to make graphics on a computer. So I think biology also uses them to model things for consciousness and other mechanisms, because it can almost make a map, an organized map, a self-organizing map of the environment and whatever inputs it's getting into the system. So that's how Julia sets work. They use kind of chaos to integrate things into the software, into the computing power, and biomolecular condensates, which are intrinsically disordered proteins. A lot of them bind sugars, and then they'll oligomerize with themselves, so that just means they'll bind to themselves. And then when they do that, they'll form a lattice structure, which can be explained through fractal patterns. And they make this different phase of water that's kind of like in between an aqueous or liquid phase and an ice or solid phase. So it's almost like a semi-dynamic structure of water, proteins, and sugar, and that makes a dynamic lattice, which can integrate things just like the Julia sets do for software. So this figure just compares a Julia set to an intrinsically disordered protein biomolecular condensate. This isn't really what it looks like. I think it's a lot more structured, but this does get the feeling that they're both disordered, and that disordered, chaotic nature of both systems gives it a computing power because it can self-organize. So this is a good diagram showing that you have a lot of different layers of code: epigenetic code, RNA code, bioelectric, and biomechanical. And one example of biomechanical would be the biomolecular condensates I talked about previously, which organize the extracellular matrix, which is the fibers, stuff like fibronectin, fibrils, or collagen, which people are more familiar with collagen, I think. So the biomolecular condensates are dynamic and work with receptors outside the cell to organize these fibers. So evolution doesn't always have to select our genetic code for the blueprint. It can also rely on these top layers of code so that it can be more robust and redundant throughout environmental perturbations. So one example of this is the flatworm or the planarian. And what you can do to these things is just chop a little piece off of them, and then they can grow back a whole new worm. Or you can even mutate the genes as much as you want, and they'll still be perfectly fine. So what that means is that they don't just rely on the genetic code for all of their reproductive purposes, and that gives them a great regenerative ability, just like a lot of these other organisms. So one way that I think this works is because the epigenetics, which is what the little methyl or acetyl modifications on the histones or DNA, all of that around the genome and how that turns different genes on and off, I think that's pretty much mapped out outside of the cell in a similar pattern throughout the bioelectricity and the morphology. So you can think about it almost like the map or structure of how the genes are turned on and off in the genome are also expressed in the structure of receptors and extracellular matrix proteins and their post-translational modifications like glycosylation, which will hold them together. So that structure is in two different places in the genome and expressed outside of the cell. So evolution can select either one to go back to as a blueprint if it has to rebuild or any other type of mechanism. So this is just showing you that bioelectrical gradient, that you have ions outside the cell, ion channels that pump ions outside and inside the cell, which is going to depolarize or polarize the cell, giving different charges inside and outside the cell, and then that's what it can use to direct cell movement and other cellular mechanisms. In the instance of the flatworm or the planarian will decide where it's going to make a head and a tail based off the bioelectric pattern by these ion channels, so they can be used to change the morphology. So I think they work hand in hand, just like I said, where if the ion channels are pumping out ions, you can think maybe those ions are going to have an effect on the biomolecular condensate, and it's going to be chaotic and be organizing things in a different way. And then that's going to alter how those fibers are moved around. And you're going to end up with a transformation of morphology at the cellular and organismal level. So I think where the magic happens is at the mesoscopic scale. I think instead of microscopic right here, it should probably be nanoscopic whenever you're looking at individual atoms and stuff. So the mesoscopic range is where quantum fluctuations meet the classical physics. And in classical physics, we see everything as a continuum. It's not as grainy as the individual atoms. So I think the magic happens in the mesoscopic scale because this is where some quantum fluctuations can actually be used to glue things together. And I'll get more on that at the end of the PowerPoint. But this is a good picture here, just showing you the different scales and the different structures you'll see at those scales. So thinking about biology from this perspective, you can think of DNA, RNA, and proteins as agents. The central dogma of biology would tell you just it's a one-way flow of information. DNA makes RNA, RNA makes protein, and then that's going to create the organism. That's the normal genetic regulatory pathway. But as I said before, there's many different software that hold this information. You also have emergent networks of agency, like the biomolecular condensates, and a bunch of different condensates can work together along with membranes to form cells, and then those cells form organisms and colonies and ecosystems. And all of these patterns can exist throughout all of these different scales, but it has the same underlying geometry, which gives a pattern recognition mechanism at these different scales, and that which can have an effect through epigenetics to the individual agents and biomolecules. And what's kind of connecting everything together is the bioelectricity, which I think, especially in humans and the brain, is where our goal-oriented behavior comes from. But I think it's a complex system in the fact that our cells have goal-oriented behavior, our organs have goal-oriented behavior, like your liver is processing stuff constantly, and our brain is just what gives us our human experience. So, as I was saying, not only cells have goal-oriented behavior, which is described by agency, but even individual proteins can have goal-oriented behavior, because one protein might operate a certain way in a mitochondria versus the nucleus versus the cytoplasm and outside the cell. So one agent, which is the protein, can have different agency depending on its localization. In a similar way that a human is going to have different behavior if they're in a classroom or if they're at home or if they're playing a sport or something like that. It's the same idea that the same agent is going to have different goal-oriented behavior depending on the context that it's in. So when you think about all molecules in the body and all cells and all tissues and all organs and all organ systems and all organisms and all ecosystems, they all have their own goal-oriented behavior. They all have their own agency. You can think how evolution can work as an operating system at all of these different scales. And you can think that there's underlying software throughout all of this. So you have environmental input. So like the ecosystem is going to dictate the environment that the genes are in and the morphology is in. So that's going to have an effect on the epigenetics and what genes are going to be expressed. And you can think about that in the sense of a software application operating system and hardware, the hardware being the DNA, operating system being the epigenetic input, and then the software applications being the morphogenesis, for instance, the biomolecular condensates I talked about before, along with the bioelectricity from the ion channels, which are held into place not only by the membrane, but by the structural proteins of the extracellular matrix, which holds the morphology or the morphogenesis into place. So, as I said before, I think the magic is happening at the mesoscopic scale. That's the scale of organelles, cells, or viruses, and also biomolecular condensates. So that's, I think, where quantum biology and classical biology meet, and that's where a lot of pattern recognition is happening. But we haven't been able to really do experiments on these patterns because of the scale that they're at. You can see cells, tissues with a light microscope, and you can see anything from molecules and atoms with an electron microscope, but trying to get to the mesoscopic scale has been really hard to do in wet tissue, and that's where we really want to see it, is when it's alive and how it's functioning. So some of the frameworks that we've made to investigate what's happening at the mesoscopic scale, for instance, the connection between a group or many different neurons, is using algebraic topology, which actually shows us that a lot of cells and like neurons in the brain, but this happens in many different cell types. The brain just happens the fastest. If you compare it to other systems and cells, they still have bioelectricity, so they still have these comparative patterns. It's just a lot slower, but they're still having this computational process. And you can see that it has a lot of seemingly Platonic geometry, which goes along with the pattern recognition ability of these systems. And I think that all goes into the fact that this Platonic geometry has a fractal-like nature, which in the same way that Julia sets are used for graphic modeling, our biology can take advantage of this so we can make kind of like a mental map of the environment, so we can kind of get an idea of what things are going to happen and have goal-oriented behavior and plan for the future. And I think this is not only, like I said, happening in the brain, but happening to other cells, and it gives them a way to compute their environment so they can have the goal-oriented behavior and function that they need. For instance, like your liver cells or any other cells in another organism. So I think this will be really cool once we really get to the bottom of this and do experiments on it and really get to the mesoscale to figure out the structural code, the pattern recognition code that is transferring at these different scales, so we can understand how these sensations of consciousness are actually happening, so that we can make robotics that have similar senses and connection to their environment and thinking behavior. And maybe if we make it good enough, we could also maybe connect our consciousness to it and start to experience how other organisms might have senses, or even download our consciousness onto a computer like you might see in sci-fi. So whenever I talk about the intersection of quantum mechanics and biology, I'm not really talking about what you might hear as Penrose hammer-off wide-scale coherence. They have their own hypothesis of how this works. And I'm also not a full classical reductionist where the quantum effects are irrelevant because everything's just kind of chaotic noise. I'm talking about that these effects are happening on the edge of chaos. So it's at the mesoscale where the quantum fluctuations are short-lived and local, but just enough to bias molecular ensembles and which are amplified throughout the system. That's why you can see that little molecular lattices, like in biomolecular condensates, can have similar structure to the the way the brain computes information will have similar structure to that little tiny molecular lattice. And I really think it probably even goes deeper than that to where the quantum fluctuations even have that similar pattern. Because after all, that's what everything is embedded in. So you would think that pattern is redundant all the way to the bottom. And that's what everything is aggregating in, which gives it a certain computational structure, which makes it very useful whenever you're doing these multi-scale computations that biological organisms do. So this also provides a route for fine individual variation or qualia, so that subjective experience. So we all have subjective experiences in our consciousness, and I think it's because it's fine-tuned, like I just said, to this level where some of the patterns in our brain can be matched to similar patterns in a fractal way to little bitty protein lattices just on the outside of some of our neurons. And then the way that that's structured is because the quantum fluctuations that kind of act as a almost like a glue to hold it together, even on the quantum scale. So that scale is repeated in a fractal way, which gives it this holographic computational power, which gives all of life the consciousness that it needs to go through consciousness-driven evolution, which I talked to on previous episodes. So I think this was a cool one, and thank you for listening, marvelous people, and I'll get working on the next one.